Hello, everyone. Thank you for joining my talk today. My name is Britney Picinic. I am a second-year master's student at York University in Toronto, Ontario. And the title of my talk today is The Effects of Diet on Aquaporin Abundance in the Disease Vector Mosquito Aedes aegypti. So Aedes aegypti is a species of mosquito, and it is located around the globe in subtropical and tropical regions. And it is actually a vector for many deadly diseases. So to name a few, there is yellow fever, dengue, and chikungunya, and a few others as well. So this makes Aedes aegypti an important animal of study because we would like to know how to control this animal by looking at its basic physiology. So Aedes aegypti female adults actually blood feed in order to produce mature eggs. So this is a very important feature of the animal and of the species. And as you can see by this image here on the screen, they're actually drinking about 2 to 3 times their body weight in blood. So it is a very large meal and something that their body has to really accommodate. And this makes them an important model for water transport because with this incoming blood meal, they have an incoming load of water that they must deal with. So blood creates this overall osmoregulatory challenge for the female. And to put it very simply, osmoregulation is the balance between water and solutes. So with an incoming blood meal, the female experiences a massive increase in water, but also in ions as well, such as sodium, potassium, and chloride. And all of these increases now need to be excreted from the body. Some are kept, of course, but the excess must be excreted. So looking at this graph here from Aquaporin in 2011, they looked at excretion following a blood meal. So indicated by the blue arrow here, we have a graph showing the fluid excretion. And as we can see at 0 minutes on the x-axis, we can see that at the y-axis, it is not at 0. Therefore, excretion of fluid has already begun before the blood meal has actually ended. So the mosquito is excreting during blood feeding. It's happening very rapidly. We also have rapid excretion of ions, such as sodium, chloride, and potassium, which are all beginning to be excreted during the blood meal, and then showing a rapid decrease following the blood meal. So the digestive and osmoregulatory organs are responsible for producing this quick effect of excretion during the blood meal. So some of the organs involved will be the following, which we have the midgut. So the midgut is the large blood-filled organ in the center of the diagram here, and it is responsible for the storage of the blood meal and for the digestion of the meal. Then at the posterior end of the animal, we are looking at the Malpighian tubules, which are the focus of my studies, and they produce the primary urine. Then we have the most posterior organ, which is the hindgut, which is responsible for the final modifications of the urine before excretion. And then we have the fat body. The fat body is not directly involved in digestion or osmoregulation. However, it is important in my studies, as you continue to see in this presentation today, and it is responsible for lipid digestion. So the Malpighian tubules, as I previously said, produce the primary urine, and they are located at the junction between the midgut and the hindgut, and there are 5 of them in particular in Aedes aegypti, with their distal end floating in the hemolymph. So if we zoom into the cells of these tubules here, we can actually see that there are 2 main cell types. So we have the principal cell here, which is the larger, more abundant cells, and the stellate cells, which are the intercalated, smaller, less abundant cells. So zooming in now further into these cells, we can actually dive into the ion transport that is occurring. So to orient ourselves, we have the principal cell on the left here, which is obviously much larger and different in structure, and we have the stellate cell here in the center. So to begin discussing ion transport, we must look at the V-type hATPase located apically at the principal cell apical membrane, which actually moves hydrogen into the tubule lumen using ATP for energy. And as this hydrogen ion concentration builds up, we have the exchange then of hydrogen back into the cell for 2 sodiums or potassiums out of the cell. And this exchange thereby creates this negative potential inside the principal cell. And this drives the secondary active transport of ions such as potassium and sodium. And then following, water is able to move via osmosis through these intermembrane channels known as aquaporins, which we will discuss. Then moving on to the stellate cell, there are a few important transporters located here. Not as many as the principal because it is much smaller. However, there is a basolaterally located NKA, which moves 2 potassiums into the cell in exchange for 3 sodiums out of the cell. And then we have a chloride shunt, which moves chloride from the hemolymph into the tubule lumen. So it moves it from one side of the cell to the other. And then as a new form of research over the last few years and continuing research, they have discovered that there is a paracellular pathway where water and chloride is able to move through the septate junctions. So this is another form of transport occurring. And overall, the goal of this is to produce an ion-rich primary urine. So now focusing on that movement of water here, we are able to see that aquaporins facilitate the movement of water and some solutes in the Malpighian tubules. Now, aquaporins are intermembrane proteins that span the entire membrane and are formed of 6 subunits, which have a hemipore in the center that is known as the NPA motif that is selective for water and some solutes to pass through. So if we look at the intermembrane aquaporins of Aedes aegypti, just looking at that Malpighian tubule diagram here, just is a very simple diagram that is just showing the cell membrane and an aquaporin here spanning the membrane that allows water to pass through. And in Aedes aegypti tubules, there are 6 known aquaporins. However, for my research and time purposes, we will only be focusing on 2 of them. So the objective of my research, therefore, shows that based on the knowledge that a blood meal poses a physiological challenge to the female Aedes aegypti that is dealt with by the production of primary urine by the Malpighian tubules, aided by the presence of aquaporins, it is hypothesized that a blood meal does in fact alter aquaporin abundance in female Aedes aegypti. So just to discuss some materials and methods of my research, I have reared mosquitoes, isolated the females, and blood fed them about 10 to 12 days after their emergence, and fixed them at timestamps of half-hour post-blood meal or 24-hour post-blood meal. And these timestamps were chosen, once again, bringing up that graph that we previously saw, based on that rapid fluid excretion that is occurring. That is why those timestamps have been chosen. And whole body immunohistochemistry has also been done, staining for Aquaporin 1 and Aquaporin 4, using custom antibodies designed for Aedes aegypti. And Western blot analysis for protein abundance in the relative organs has been done and is the subject of continuing research for myself. Now for adult female Aquaporin 1 localization, here we are looking at the posterior end of the animal. And to begin with the Malpighian tubules, we see that Aquaporin 1 is apically located and shows fairly dim staining in the tubules. Moving on to half-hour post-blood meal and 24-hour post-blood meal, we see a significant increase in brightness of Aquaporin 1 abundance. If we look at the fat body now, we see that in the non-blood fed, Aquaporin 1 stains very bright. And then in the half-hour and 24-hour post-blood meal, the staining of the fat body has decreased significantly. Now, if we look at the effects of the blood meal and Aquaporin 1 abundance in the tubules using a Western blot, so once again, we are looking at the posterior end of the animal here, the tubules, and this down here is a study by Drake and colleagues in 2010 that is a basis for my research as well, showing that mRNA levels of Aquaporin 1 actually significantly increase about 3 hours post-blood meal. So based on that timestamp and their research, we looked at half-hour and 24 hours. And now our Western showed that after analyzing these bands here, there was actually a significant decrease in the 24-hour post-blood meal group. That was the only significant change, which actually goes against our immunohistochemistry as well. So this is the subject of continuing work for myself, which is being now looked at on more of a subcellular localization in terms of cytosol versus cell membrane. So that is the subject of continuing research for me. Then if we look at the effect of the blood meal on aquaporin 1 abundance in the fat body, so based on this research as well, so Drake and colleagues did not find any significant differences in mRNA levels with a blood meal. So now analyzing these bands here, we also see no significant changes in the post-blood meal groups. So now looking at aquaporin 4 localization in this adult female, once again, still at the posterior end of the animal, we see that in the Malpighian tubules, the apical staining is fairly dim. We see that same thing in the half-hour post-blood meal, apical staining is fairly dim. However, in the 24-hour post-blood meal group, the apical staining is significantly brighter. Then the fat bodies show the same trend as before, where they are the brightest in the non-blood fed compared to the blood fed groups. So the effect of the blood meal on aquaporin 4 abundance now in the fat body has shown by analyzing these bands that there is no significant decrease in the aquaporin 4 abundance. However, there is a decrease in the 24-hour post-blood meal group. So this will be repeated with more replicates to hopefully produce a more accurate result. Now, skipping away from the females for a moment, I just wanted to touch on some new work that we are continuing to do, and it is now looking at the effects of a diet on the male Aedes aegypti. So male Aedes aegypti require a nectar meal in order for them to survive, and they do not blood feed, so it is unique to the females of the species. And my work will focus on sugar-fed males, so their normal diet, versus starved males, so given no meal at all. And so far, looking at aquaporin 4 abundance with a Western here, we see that in the starved group, there is a significant decrease in the protein abundance of aquaporin 4, which is very interesting and something we will continue to pursue. So in conclusion, immunohistochemistry provides evidence that aquaporin localization may be altered by a blood meal, specifically in the Malpighian tubules and fat body. Western blot analysis provides evidence that the protein abundance may also be altered by a blood meal. And Western blot analysis of the male fat body indicates that their nectar-based diet may affect aquaporin abundance as well. So some continuing work will be focusing on hormonal control of aquaporins, in tied to the blood feeding and effects of diet. So that will be very interesting and the subject of my research. So thank you all so much for listening today. Thank you to the members of my labs, the Danini Lab, Paluzzi Lab, and Kelly Lab. And thank you so much, BSI, for the opportunity to participate. Thank you all so much.